Good morning, everyone. I'm Daniel Tennant. I'll be one of the co-chairs for the morning session. Without further ado, I'd like to introduce our keynote speaker, Carlo Croce. So, like most supermodels and rock stars, Carlo is a first name only rock star. But one thing I want to say seriously about Carlo, I always admire people who have multiple careers in science. I'm afraid to say I'm not smart enough or talented enough to have multiple careers, but Carlo really has. I grew up actually reading studies of his on translocations in MYC and probably a lot of things that I've forgot. But in recent years, he's really been a leader in the field of microRNAs in cancer. And I think it's quite remarkable for someone to take a new field and really lead it. And so today, I think the subject of the talk will be continuing in that vein, causes and consequences of microRNA dysregulation in cancer.
Thank you, David. I want to thank the organizer of this beautiful meeting. It is really a great pleasure to be here. During the next 35 minutes or so, I will tell you about this long journey that brought us to the discovery of the involvement of a new class of genes that do not encode for protein, in the pathogenesis of human cancer. And I hope to convince you that, in fact, microRNA dysregulation contributes to the pathogenesis of every malignancy. So, as David said, we started this long journey by trying to figure out the role of genetic alteration in the pathogenesis of cancer, and we started this journey studying a malignancy. This work was done with Riccardo Dalla Favera a long time ago. We tried to figure out whether specific genetic alterations are involved in cancer pathogenesis. We started with Burkitt lymphoma. And what we found nothing, apparently. Can we move forward? And we found now it works? Fine. We found that in Burkitt lymphoma, there is a juxtaposition of the human homologue of a retroviral oncogene called MYC to the three immunoglobulin loci. The most common alteration is the 814. And because of this translocation, the MYC gene involved in this reciprocal translocation is dysregulated and is expressed constitutively at elevated level, and that is very likely the first genetic alteration. So it's the initiating event that will result in malignancy. And then we thought that we could exploit highly consistent chromosomal alteration to clone cancer genes just on the basis of their chromosomal position. And after this, we cloned the BCL1 locus that turned out to be cyclic D1 that is involved, in fact, is the initiating event in the pathogenesis of mantle cell lymphoma. And immediately after that, Yoshida Tsushimoto, working in my lab, cloned this gene that became enormously popular that is involved in follicular lymphoma. This is the initial event in the pathogenesis of follicular lymphoma that I name BCL2 for B-cell leukemia lymphoma 2. And later on, I will talk a little bit about this gene. And then we continue to use this approach to clone and characterize genes which are involved in the initiation of malignant transformation. And we clone by this approach both oncogene and tumor suppressor gene. Another gene very popular that we clone by using this approach, Gisele Canani, was the gene that we call ALL1, that changed the name later on as called MLL. Now, more recently, I was interested in figuring out the molecular mechanisms which are involved in the pathogenesis of the most common human leukemia, which is chronic lymphocytic leukemia. As you know, this is the malignancy of CD5 positive B cell, and can present itself in two forms. In an indolent form, and patients with indolent CLL often die with this CLL and not because of this CLL. and can appear as an aggressive form. And when a patient has an aggressive form of CLL, the patient has very poor, will survive for another 2 or 3 years. So we look at the cytogenetic result done by many different laboratories concerning CLL, and also in the case of this disease, some consistent chromosomal alterations were described. The most common is this alteration, a 13q14 deletion, which is observed by cytogenetics in 55% of the cases. Now we know molecularly this alteration occurs in about 70% of the cases and occurs in the indolent form of CLL. The second most common alteration is the deletion of 11q23 that occurs in 18% of the cases. This occurs only in the aggressive form of CLL. The third most common alteration is the trisomy of chromosome 12 that occurs in 12 to 14% of CLL. And finally, the fourth most common alteration is the deletion of 17p, where many years ago we mapped the gene for p53. So we decided to focus on the most common alteration, which is the 13q14 deletion. But we realized immediately that these deletions are very, very large. And therefore, we have used an approach developed by Webb Cavani many years ago called loss of heterozygosity to narrow the region of interest in order to find the gene. And so we started this very large LOH study trying to find precisely where the putative suppressor gene involved in CLL is located. While we were trying to find the CLL gene, I was also trying to find the gene that is responsible for a rather uncommon T-cell malignancy called T-cell prolymphocytic leukemia. And in 98% of T-cell prolymphocytic leukemia, there is either this rearrangement or this rearrangement. This is a 14-14 translocation. This is an inversion of chromosome 14. And many years ago, we mapped the alpha-delta locus of the T-cell receptor here on chromosome 14. And therefore, because this, the breakpoint, one of the two breakpoints in both the translocation and the inversion map precisely here, we thought that, in fact, the alpha-delta locus of the T-cell receptor was involved, and then we found that, in fact, that was the case. And then Gianni Russo in my lab, walking away from the alpha-delta locus of the T-cell receptor, found this gene that I named TCL1 that is involved in the pathogenesis of T-cell prolymphocytic leukemia. In order, this gene encodes a protein 114 amino acid, which is present in the nuclei and cytoplasm of early B and T cell, is for the most part an early lymphoid-specific gene. And then in order to prove that TCL1 is really the gene involved in T-cell prolymphocytic leukemia, we made a mouse model in which we dysregulated the human TCL1 open reading frame by using the LCK promoter enhancer element, and this mice at the average age of 15 months developed T-cell prolymphocytic leukemia, proving that, in fact, the dysregulation of TCL1 is responsible for this disease, both in human and in the mouse. But then Gianni found something else quite interesting. He found that TCL1 is also expressed at high level in a variety of human B-cell malignancy in which TCL1 should not be expressed, because TCL1 is not expressed, or is expressed at very low level in mature B-cell. And therefore, we asked the question, what would happen if we dysregulate TCL1 in B-cell? And so we used this construct to make a mouse that now became very famous. It's called the immune mouse. And we found that this mice express the human TCL1 open protein in their lymphoid tissue. The non-transgenic did not. In the liver, where the enhancer doesn't work, there was no expression. So we got mice in which there was an overexpression of TCL1 in all their B cells. And we observed something very interesting in this mice already at 2-3 months after birth, because we found an expansion of CD5 positive surface IgM positive B cell, which, by the way, is the phenotype of CLL cell. And then, at the average age of 12 months, these mice, as the penetrance of 100%, develop a CLL. Okay? You see large spleen, large liver, large lymph node. So this regulation of TCL1 can cause a CLL. But I told you at the beginning of my talk that in human we see two kinds of CLL, the aggressive form and the indolent form. So what kind of CLL do these mice develop? These mice develop only the aggressive form of CLL that made us to think that perhaps this regulation of TCL1 or of the pathway that involves the TCL1 is involved in the causation of the aggressive form of CLL. Then we tried to figure out how TCL1 works. We found that in this work of Yuri Pekarsky in my lab, we found that TCL1 is an activator of AKT. You know, AKT is the human homologue of a retroviral oncogene called AKT8 that induced T-cell leukemia in mice. So, and we are convinced that this pathway is involved in the pathogenesis of the T-cell proliferative leukemia. But on this side, there is a problem, the B-cell side, because we made, in collaboration with the field cyclist, mice in which we dysregulated AKT1, AKT2, AKT3 in B-cell, and these mice did not develop B-cell malignancy. And in fact, the P10 knockout doesn't develop a B-cell malignancy. And then, to make a long story short, Yuri Pekarsky found that TCL1 is an activator of an FKB pathway and also a strong repressor of AP1 transcription complex. At this point, we sequenced TCL1 in 600 CLL, and we found 3 cases in which there was a gain-of-function mutation of TCL1, and they were, all these 3 were very strong inhibitors of AP1. And the more recent, I will tell you, we found a third function of TCL1. Then let's go back to our loss of heterozygosity study. We, and also Ricardo Della Favare's lab and others, found that this is really the critical region which is involved in loss in CLL. And at that point, there was no complete sequencing of the human genome, so we sequenced 2 megabases of DNA with this in the middle. This was a long time ago. And then we asked a very simple question. Is any gene contained in this region specifically altered in CLL? We worked on this project for 7 years. We couldn't find it. And in fact, my senior postdoc, who was involved in this study, decided to get an MBA from the Wharton School of Business and left science altogether. So this was a very distressing time. I thought that the gene had to be there, so I thought that maybe I could find where the gene was by using a rare translocation. So from MD Anderson, from one of my collaborators, I got a CLL with a rare translocation between chromosome 2 and chromosome 13, and George Cullen in my lab mapped precisely the breakpoint in this leukemia with a beautiful single cut here. You see, it involved the critical region, but there was no gene there. And then the definitive clue came from the father figure of CLL research, Kantirai, that one day at the meeting told me, Carlo, I saw a very interesting case of CLL, and I asked why interesting, because it's in a patient with familial retinoblastoma. The gene for retinoblastoma is also SERTINQ14, but is on the left. It's not involved in CLL. So I thought that perhaps that patient had the large deletion that knock out the retinoblastoma gene and the CLL gene. That turned out not to be the case. But then I got the cell with my own hand. I segregated the 2 chromosomes 13. That tells you that once in a while I still work in the lab. And in one of these 2 chromosomes 13, we found the clue, because we found that this patient had a deletion precisely in the same region. You see overlapped with the translocation breakpoint, and the deletion was only 31.4 kilobases, but there was no gene there. Fortunately, once in a while, I still read the literature, and I started reading about microRNA gene. You know, the first microRNA gene was discovered, and remember this, by Victor Ambros in 1993 in C. elegans in the world. And mutation in this gene called LIN4 generate a defect in the development of C. elegans. After that, there was absolutely no interest in microRNA whatsoever. Only after 1998, when siRNA were found, and they seemed to be working in a similar fashion, there was a renewed interest in microRNA genes. More microRNA genes were discovered. The Let's Set family of microRNA was discovered. In year 2000, it was found that Drosophila contains microRNA genes. And then in 2001, it was found that rats, mice, and humans have microRNA genes. And we were in 2001. So I called George Karin, and I asked George, look at whether in this region there are some microRNA genes. I still remember his face when he came to my office, and he dragged me to his computer, and he found me this, that in fact, at breakpoint, there were two microRNA genes, miR15 and 16. And so it became very clear to us that loss of miR15 and 16 must be the critical event in the pathogenesis of the indolent form of CLL. And we went on, we analyzed over 50 cases of CLL at that time, and we found that miR15 and 16 are knocked out or knocked down in about 70% of CLL. Okay? So loss of two microRNA is the initiating event in the pathogenesis of the indolent form of CLL. I don't have time to talk about ART. At this point, we were convinced that microRNA are involved in the pathogenesis of human cancer, and therefore we map all the known microRNA genes, and we got another surprise because we found that several microRNA genes map precisely to regions of the genome which are involved in consistent chromosomal alteration in a variety of human cancer. And I hope to convince you at the end of my talk that in fact this regulation of microRNA occurs in all human cancer. You know, microRNA are regulator of gene expression, so they are transcribed by RNA polymerase II. A large trans is processed by DROGA to this, which is called primer. After DROGA cleavage, you have the formation of primer that bind S protein 5, go to the cytoplasm where you encounter dicer that cut off this, and then this double-stranded microRNA that in general between 21 and 22 nucleotides in length unwind, and then the mature strand is incorporated in the RISC complex, and this short RNA molecule bind by partial complementarity, for the most part, to the 3' region of messenger RNA. And when that occurs, two things happen at the same happen. There is a blocked translation and or degradation of the target RNA. So microRNA are in fact a regulator of gene expression. And later on, at the end of my talk, I will tell you that microRNA are also an additional function. And microRNA can be suppressor, like for example miR15 and 16, or they can be oncogene, like for example miR155, which is amplified in the ABC type of diffuse large B-cell lymphoma. But you should not make the mistake that many people make to call a microRNA an oncogene or a chemosuppressor gene, because everything depends on cellular context. In fact, I will give you some examples of microRNA genes that are oncogenic in one cell type and suppressors in a different cell type. So some microRNA, so miR15 and 16 function as a suppressor. You can have homozygous deletion of one allele, and promoted methylation of the second. I will show you the first cases of, that we found, in fact, of the mutation in microRNA genes. So in this case, the microRNA function as suppressor. You can have amplification at the DNA level, for example, miR155, that in fact seem to behave like an oncogene. And this is the very interesting case. In a long time ago, as you can see in this paper, we cloned a chromosome 817 breakpoint, and we found in an acute lymphoblastic leukemia, and we found that that translocation led to a fuse gene and led to the fusion of MYC gene to a gene on chromosome 17 that we call BCL3. Okay? And we found that BCL3 is transcribed into a 1.7 kilobase message, which is expressed at very high level, in fact, in all hematopoietic lineage. Then, unfortunately, we sequenced it, and we found that it did not have an open reading frame, and we dropped the project. That is the precursor for miR-142, which is the most highly overexpressed microRNA in all hematopoietic lineage. So that you see immediately how dogmas are very negative for science, because that finding did not make us to continue that project and find, in fact, miR-142. At this point, we developed the first chip to look at global expression of microRNA genes in tissue, and this was our first result that indicate that the pattern of expression of microRNA is absolutely tissue-specific and cell-type specific. You can tell the cell, you can tell the tissue just by looking at microRNA expression. So we went back to study CLL. These are about 50 CLL that were analyzed by our chip. And you see, in fact, that there were two different groups of CLL, the group on the left, the group on the right. This corresponds to the indolent CLL, this corresponds to the aggressive form of CLL. So by using this approach, you can make the correct diagnosis of a human malignancy. We have extended this observation in a paper that we published in New England Journal of Medicine, where we added the 94 more cases of CLL to our analysis. We found a small signature represented by 13 microRNA that allow us to define the behavior of the CLL, whether it was indolent or aggressive. At this point, we sequenced 42 microRNA, including the 13 in the signature, and we got another big surprise, because we identified the first mutation in microRNA gene. Naturally, at that time, we were mostly interested in this, so we sequenced this 42 microRNA in 75 CLL and 160 normal, and you see that we found two cases of CLL in which there was a mutation, 7 nucleotide 3' of miR-16-1. Remember that miR-15A and 61 are on the same polycistronic RNA. And this mutation affects the processing by droga of the precursor. So there is less mature microRNA made. Interestingly, we found that in these two patients, in the leukemic cell, we could find only the mutated allele. That seems very strange to us, so we look at the buccal mucosa of these two patients, and we found that they were heterozygous. So their leukemic B cell have lost the normal miR-15, 16 allele, fulfilling the Knudson two-hit hypothesis for tumor suppressor. Then we found mutations, some somatic, some germline, in other microRNA genes. But you will notice immediately that the frequency of this mutation is very low. In fact, the mutation involving the miR-15, 16 cluster occurs in about 3% of CLL. Interestingly, one of these two patients, a female, previously developed breast cancer. Her mother died of CLL, so it was a CLL family, and her sister died of breast cancer. So perhaps in the context of this family, but this is not proven at all, this germline mutation might have affected also the risk of developing breast cancer. So this are the position of this mutation that we described in the New England paper. Interestingly, Elizabeth Ravici recently was interested in figuring out the genetics of an indolent CLL that appear in one mouse strain, NZB. The NZB mice are the only mice that develop an indolent form of CLL late in life, like in humans. And she mapped this disease to mouse chromosome 14 in a region that is homologous to 13q14 in humans. So she thought there could be miR-15 and 16 mutation. So she sequenced it, and in fact, she found only in the NZB mouse strain a mutation that was not 7. but 6 nucleotide 3' of miR16-1, and that also affects the processing by Drogen. So germline mutation of miR15 and 16 caused the indolent form of CLL both in human and mouse, and more recently Ricardo Dalla Favara's lab published the knockout in cancer cells of miR15 and 16. Also, those mice developed the indolent form of CLL. Then we were interested in target. And you know that there are several algorithms that you use to predict target, and this algorithm suggested that BCL2 could be a target. And I found it very interesting because in human, you know that there are two major indolent B-cell malignancies. One is follicular lymphoma, and we know that it's caused by the dysregulation of BCL2 because of chromosomal translocation, the 14-18 translocation, and the other is CLL. So in CLL, if miR15 and 16 target BCL2, BCL2 should be high in CLL, and it is. In fact, there is an inverse correlation between miR15, 16 expression and BCL2. And then we went on to prove that, in fact, the BCL2 is a target of miR15 and 16. In this case, we look at the human leukemic cell line with biallelic deletion of miR15 and 16. So we put in a vector the wild type miR15 and 16, and the mutant miR15 and 16 we got from one of those two patients. See, when we put the mutant here, you see, this is a Western blot, you see a decrease in the expression of BCL2 and no consequence. When you put the wild type, there is a beautiful knockdown of BCL2, activation of APAF1, activation of Caspase-9, degradation of PARP, and apoptotic ladder. And which indicate that by re-expressing miR15 and 16 that was lost in the leukemic cell, you killed them, which suggests quite strongly that miR15 and 16 could be drugged in the treatment of tumors which are driven by BCL2 dysregulation. Then what about the aggressive form of CLL? So these are 8 CLL, 6 aggressive and 2 indolent. You see that the indolent express a little bit of TCL1, but not very much. The aggressive express a lot of TCL1, and to make a long story short, we found that at least in part, this high level of expression of TCL1 is due to the loss of microRNA that target TCL1, particularly miR181B. So also a similar mechanism that occurs in the indolent form of CLL occur in the aggressive form of CLL. This is the additional function we found for TCL1. This is work of Yuri Pekarsky. I don't have time to discuss it. We found that TCL1 is a very powerful inhibitor of the de novo DNA methyltransferases. So it causes generalized hypomethylation. So, and you know that mutation in the DNA methyltransferase 3A, and later on they were found in also the 3B, occur in acute myelogenous leukemia. So this global hypomethylation we found not only in the aggressive form of CLL, but also in our TCL1 mouse. Then we wanted to figure out whether there is an interaction between the different type of genetic alteration that we found in CLL. You know that a biomarker for aggressive or indolent CLL is a protein called ZAP70. It is high in aggressive CLL and is low in indolent CLL, and nobody knows why. Okay? This is why. We found, this is the work that we just published in JAMA about a year and a half ago. We found that miR15 and 16, which are a certain Q1 form, are lost because of the deletion. We found that they not only regulate BCL2, but they also regulate MCL1, another anti-apoptotic gene that is overexpressed in CLL. So when you lose these two, you have high BCL2 and high MCL1. And that is probably one of the problems with the ABT drug in the treatment of CLL, because the CLL not only overexpress BCL2, but also overexpress MCL1, and the Abbott drug doesn't shut down MCL1. But we also find that p53 is a positive regulator on miR15 and 16, and these two miR regulate p53. So if you use genotoxic drug or irradiation, and you cause an upswing of the expression of p53, this will lead to overexpression of miR15 and 16, very high level of expression of miR15 and 16, that will shut down these two anti-apoptotic genes, and the cell will die. Now, p53 is also a positive regulator of 3 microRNA of the miR34 family, miR34A, B, and C. miR34B and C are on 11q23, and they are always deleted in CLL with 13q14 deletion. Okay? So whether you lose p53 or you lose 11q23, these two are knockout. And we found that these two microRNA regulate negatively ZAP70. So that is the reason why ZAP70 is overexpressed in CLL with p53 deletion or mutation and with 11q23 deletion. So some microRNA function as suppressor, some function as oncogene, but as I mentioned to you, everything depends on cellular context. For example, look at this. miR221, 222. They are lost in erythroblastic leukemia. You put them back in erythroblastic leukemia, you kill the leukemic cell. And the critical targets seem to be KIT. So they are negative regulator of KIT. They are gone, KIT is high, and they contribute to the malignancy. But miR221, 222 are the most dysregulated microRNA in hepatocellular carcinoma. The level of miR221, 222 in metastatic and invasive hepatocellular carcinoma can be 30-50-fold the normal level. And this microRNA target a number of tumor suppressor genes. In fact, we show that they target P27, P57, P10, and TIM3. So overexpression of these two microRNA result in shutting down 4 major tumor suppressor genes at the same time, and that contribute to the pathogenesis of hepatocellular carcinoma. Now we have proven this point by making mice in which we targeted the overexpression of miR221 into the liver of mice, and these mice developed hepatocellular carcinoma, and we could cause the regression of this tumor in this mice by injecting intravenously an anti-miR221 into this mice. So this suggests that at some point we could use anti-microRNA treatment, at least for the treatment of this very common tumor. So that we don't have time to discuss this, but we have time to discuss it. In order to prove that, that a single microRNA can cause cancer, we decided to dysregulate miR155 in B cells since, as I mentioned to you, the BC type of diffuse large B-cell lymphoma have an amplified miR155. So we dysregulated miR155 by using the new enhancer element of immunoglobulin. We got extremely high level of expression of miR155 in the B cell of these mice, and this is what happened. These are two mice. They are 2 months old. One is wild type, one is transgenic. There is any difference? One seems to be fat, but it's not fat. It has a huge spleen. And in fact, and then you look at the spleen, A, B, C are 3 spleen of 3 mice, 2, 3, 4 months old. The structure of the spleen has been destroyed by leukemic cells. This is the bone marrow of the normal, of the wild-type mouse. This is the bone marrow of the transgenic mouse, and this cell stained positive for Ki67. So this might develop an acute lymphoblastic leukemia, and they are all dead within 6 months. So this microRNA work as efficiently as MYC in inducing an extremely aggressive B-cell malignancy. These are the culprits. They are large pre-B cells. Now I don't have more time to tell you about treatment of mice with miR15 and 16, but I want to make a point before I close. After this work, we established signature for the distinguished tumor tissue from normal tissue for many different solid tumors. And then we asked the question whether there are commonality in microRNA dysregulation about these different tumors. And at our early study that we published a few years ago, we studied just 6 different solid tumors, some of the most common human tumors. Now we have extended this to 15 different solid tumors. And we asked whether there are commonality. And in fact, there are. And in fact, as you can see, miR-21 is overexpressed in 6 out of 6 solid malignancies. In fact, it's overexpressed in 15 out of 15 solid malignancies, and so on. What does it mean? It means something extremely simple. It means that some of these microRNA are in fact downstream targets of pathways which are commonly involved in cancer pathogenesis. That is the reason why microRNA are always dysregulated in every human malignancy. Okay? Because some of them are downstream targets of pathways which are involved in cancer pathogenesis. And that is the reason why they are dysregulated. Then we found something else, and that is that some of the microRNA affect the repair. We found that miR-155, miR-21 regulate mismatch repair genes. So to make a long story short, the microRNA like miR-155 and miR-21 not only shut down tumor suppressor, but they generate a mutator phenotype. So the frequency of mutation increases because this microRNA shut down repair. Okay? So this certainly contributes to mutation rate and to the pathogenesis and progression of malignancy. Finally, I want to close with this slide. We found that microRNA have also another function. They are not only a regulator of gene expression, but they are also hormones. We found, and we just published it a month ago, that the cancer cell, we have done it with lung cancer cell, but it's also true with breast cancer and with colorectal cancer. We found that the cancer cells secrete microvesicle or exosome containing microRNA. This microvesicle fuse with cell of the microenvironment. And the load, the microRNA load, end up in the cytoplasm of the cell, of the microenvironment. Some microRNA reach the endosome, and some microRNA, based on their sequence, bind specifically to toll-like receptor 8 in human and toll-like receptor 7 in the mouse, which is the homologous of TLR80 in human. And they are agonists of toll-like receptor, and that results in the induction of tumor necrosis factor alpha and interleukin-6, and that enhance the ability of the cell to invade and to metastasize. So in summary, this regulation of microRNA occurs in every tumor and contributes to the pathogenesis of every tumor. And assessing the dysregulation of microRNA can be used for better diagnostic, prognostic, and hopefully, I think, for therapy of human cancer. Thank you very much for your attention.
Very impressive work, especially I started with MYC. You know, that's one of the earliest cloned genes. It turned out so important. Even for IPS becomes indispensable. So without MYC, there won't be IPS, and there won't be a Nobel Prize of this year. I just wonder, so you have done this biology, cancer biology getting more and more complex. We studied genes, protein, now we studied the microRNA, and then there will be 95% non-coding RNAs there. I wonder, probably one way we want to see is to find the common features. And you have found, you have reported that microRNA functional similar on many aspects, but anything about the structural, I mean the translocation. I like your current translocations. Ever try to look at the sequence similarity or see elements around the translocation regions in cancer?
Okay, so naturally I love translocation because we also often use them to clone genes. Naturally, before we thought the translocation were involved in cancer pathogenesis only in leukemia cell lymphomas. Although it was clear that the mechanism of translocation in B-cell and T-cell malignancy for the most part involved the same enzymatic system which is involved in immunoglobulin and T-cell receptor gene rearrangement, is very clear now. We did not know what was the mechanism involved in myeloid tumor, but clearly there are mechanisms, enzymatic mechanisms in the cell that allows this translocation. Now we see an explosion of translocation, although they are not that common in solid tumor. For example, the ALK translocation in lung cancer, the translocation of the S family gene in prostate cancer. So also solid tumor can involve translocation, although we don't know yet whether the translocation in the solid malignancy occur during tumor progression or are initiating events. I suspect that in many cases they are progression events and not initiation events. But time will tell. It's becoming clear that this mechanism, translocation or inversion and so on, is also common in solid tumor. So thank you very much for your comment.
So I have two questions. First, how common are alteration in the 3 UTRs?
In the
3 UTRs of target genes of microRNAs in cancer? That's one question. And the second question, while there are oncogenic microRNA, quite curiously, in many cancers, for example, ovarian cancer, there is general downregulation of the mRNA processing machinery, like shut down of all microRNAs. How do you explain this?
As I mentioned to you, that's a very good question. As I mentioned to you, you have to look at cellular context, okay? And there was a paper in Nature published many years ago by an Harvard MIT group that claimed that microRNA genes are always downregulated in cancer. Don't believe it because it's not true. Some microRNA are up and some microRNA are down. I have never seen a cancer where all the microRNA are down. But some of those are critical, okay? And are critical in that context. So what it makes this story fascinating is the fact that microRNA, at least certain microRNA, cannot be dissociated from the pathway that regulates them. They are part of the pathway, okay? For example, if I activate EGF receptor, I can read that dysregulation through dysregulation of few microRNA. If I dysregulate MET, for example, I see immediately MIR221, 2 going up. If I dysregulate MEC, I see the MIR1792 cluster going up, I see MIR29 going down, MIR26 going down, and so on. But dysregulation of some of these microRNA is critical. It's the way the pathway works, okay? So that is the reason why we get hope to target the entire pathway by using microRNA or anti-microRNA, because the dysregulation of those microRNA contribute to pathogenesis. I want to give you an example. Josh Mandel has published in Cell a couple of years ago a paper where he studied in the mouse tumors that were induced by MYC, transgenic MYC mice, where dysregulation occurred in the liver. Okay? And he found that all the targets of MYC were dysregulated. Okay? But many microRNA were dysregulated. Okay? Many, including MIR26, that was downregulated. He made an AAV vector containing MIR26, so only one of the MIR dysregulated by MYC infected the liver of this mice, the tumor regressed, okay? Which indicates that we can really take advantage of the fact that microRNA are downstream target, and microRNA are always targetable to induce the regression of the tumor. So I think that microRNA and anti-microRNA will be drugged. The problem is targeting. Targeting to liver is no problem because you can get it spontaneously by injecting the microRNA by intravenous. Okay? For tumor, the pancreas and so on, you will have to figure out a way to target the microRNA to the tumor.
So it looks like Sam will interrupt with one burning question, then Shai, if you want, you can come back in a second.
My question's going to be short, but I don't know about the answer. So Carlo, what's known about microRNA changes with age, and is it any way related? It's probably too long.
This is a very important question, I think.
And secondly, do silence genes induce microRNAs?
So, microRNA change during aging process. What I have done, and I have not published it yet, but I have done it looking at skin fibroblast and also at WI38 of different passages. And it's clear, for example, that some microRNA go down. Particularly, I can tell you, MIR221, okay? MIR221 goes down, okay? Now, it would be exciting to see whether we can rejuvenate a little bit this cell by continuous expression of this microRNA in the cell. But there is no question that microRNA change during the aging process. And you ask also another question? Oh, if the gene is silenced, it depends. So if let's say, the pathway or the transcription factor that upregulate microRNA is silenced, those microRNA will not be expressed. If it's the negative regulator of those microRNA, the microRNA will jump up. Okay? And in fact, we are studying now very extensively several cases in which the transcription factor is lost, or there is a loss of function of the transcription factor, and the microRNA target goes up. Okay? So you cannot dissociate the pathway in our traditional sense from microRNA because they are part of the pathway.
Okay, we'd like to thank Carlo for a great talk.